plasmid sequencing was a real game changer when it comes to seeing if your molecular cloning or site-directed mutagenesis worked. And not only did it insert the gene that you wanted, or did it insert the mutation that you wanted, but there aren't mutations elsewhere in the plasmid. Before, you would typically use Sanger sequencing. With Sanger sequencing, here you make lots of copies of the DNA, and as you're adding the DNA letters, well, those DNA letters were actually going to be fluorescent, and then you could read out the sequence kind of as it's getting made or after it gets made. But you could only do these short regions, and you had to define the regions because that tells DNA polymerase where to go. And so typically you would design your primers, or you would use primer sites that were like common to the plasmid in order to sequence the region of your gene or of your insert. But you might not actually have a long enough read to cover that gene for that region. And you're then missing all the other places or in the plasmid where there could be unintended mutations. And so those mutations could be impacting whether the protein gets made correctly and things, but you're not going to know because you're not looking there. And if you want to look at the rest of the plasmid, you had to actually go and use multiple primer pairs and get multiple reads. So typically, though, you would just do kind of the region of interest, but you had to design your primers knowing that the beginning and the end of those reads were not going to be reliable. And so it was a big pain. Enter whole plasmid sequencing. With whole plasmid sequencing, there's no primers or anything. They sequence the entire plasmid. So, how's it done? So, the version I use at least uses Oxford Nanopore Technologies, or ONT. Basically, nanopore, what it is it's literally these tiny little pores in this membrane, and you thread DNA through those pores, and as the DNA is getting threaded through, it's going to kind of give a signal that's going to allow it to be read out, or in the bases called, so basically seeing which nucleotide is going through when. The way that this is able to be done is that there's also ions flowing through there, so charged molecules. And we talk about the movement of ions, booyah, you've got electricity. If DNA is also flowing through that channel, it's going to impact the movement of those ions, so there's going to be differences in the electric current. And there's going to be differences depending on which nucleotide is actually going through right at that time. Is it an A? Is it a G? Is it a C? Is it a T? And it can even sequence RNA. And it can detect modifications to DNA and RNA. So lots of cool stuff. What's also really cool is it can do really long reads. It doesn't need the DNA polymerase. So it needs like this adapter that's going to have a protein that is going to thread the DNA through the pore. So a motor protein. It's kind of like unwinding the strand, unzipping the DNA so the DNA can go through the pore, and as the DNA is going through the pore, it's going to impact the flow of the ions, which is going to be then like read out, and you can call the base. But the plasmid is circular. You need a way to get it to be linear, and you need to thread it through. How they do this is they use this, well, there's different methods, but this method uses this kind of like transposase. And so the transposase is this enzyme that is going to basically cut the DNA and add on a unique barcode. So a sequence that is specific to that sample. And this allows for things like multiplexing if you want to mix samples together. Well, now you know that the ends of the sequence kind of have this unique part that is specific to that sample. The transposome that they use basically has fairly low preference in terms of where it cuts. So you're going to get cuts at multiple places around the plasmid, and each of those places you're kind of linearizing it in a different way, and you're adding those barcodes onto the end. And so you're kind of going to cover the plasmid. Each read should hopefully be long enough because these can do such long reads that they can typically get all the way around the plasmid. But if they can't, no biggie, because you're cutting it in multiple random places around. And so these reads then just have to be aligned afterwards, and you can see the regions line up, and then you can kind of get high coverage of each region. Now, how are you going to actually get it through though? On the barcodes, those are also serving to hook up to sequencing adapters. And those adapters then have a motor protein that's going to be doing the unzipping of the DNA and helping it get through the pore. And so those are going to hook onto those unique barcode places, and those unique barcode places were going to be added by the transposase, which kind of acts like scissors that stick the sequence onto the ends, and then they use chemistry to actually attach the sequencing adapters to those, and those sequencing adapters are then going to allow the DNA to get threaded through the pores. and it's going to then give that readout that is then read as a base, and then you can align the reads and you get high depth coverage of various regions. And so you can kind of, if there's a mutation or like a mistake with the read, not a mutation in the DNA, but actually just a mistake in the read, a mistake in call, then what can happen is that you can say, okay, well, I know from all of these other ones that this location or this letter, and this is saying it's this letter, this read's probably not great. And so that allows you to make up for errors in this processing when you're doing these long reads, is having that high coverage. And you're going to typically have high coverage of hopefully all the areas, because what happens is that you're cutting it at different places. And so you don't have to make copies though. So unlike other methods like Sanger sequencing, where you're actually having to make copies of the DNA, and methods like Illumina and things where you're often making copies before you actually go and sequence it, and then when you're sequencing it, here there's no copying, and so you don't introduce the mutations in that way. You don't introduce the read errors in that way. And instead you just have to worry about kind of the accuracy of the reads as they're being kind of like called. But it's really cool, and it's not even like it's more expensive really, and I'm just a big fan of whole plasmid sequencing, and I'm also a fan of the fact that my students' cloning worked, and my lab student, Karina, shout out to Karina, she got 4 great clones, and so we're just ecstatic, and it was a hard-won effort, and we got it.